Okay, chapter 28 is about the reproductive system. I think you guys are already familiar with these models, but it's a good opportunity to understand lecture, and also if you see questions in the lab, you can answer it from here. So pay attention. So we're basically going to discuss male reproductive, female reproductive, internal and external. How do we make the sperms? How we make the eggs? And the different glands and secondary sexual characteristics, primary sexual characteristics, and so on. So the reproductive system, the main thing is, why do we have reproductive system? We have it for making the gametes, right? And for sexual intercourse and for pregnancy and having kids, right? These are the basics. So the general terms that you need to know are gonads and gametes. Gonad is a general term that means testes or ovaries. Both of them are called gonads, right? When you say gonads, you're not being specific. Gonad can be testes or ovaries. Gametes, same thing. You're not being specific. What do you mean by gametes? Is it male or female? If it is male, it is, what's a male gamete? Spermatozoa or a sperm, same thing. And what is the female gamete? It's a oocyte or the ovum. Okay, there are internal organs and external organs. We are going to cover that. And obviously, male and female reproductive systems are different because of the pregnancy. Basically, this is the main difference between males and females. Um, and the other thing is, because of the pregnancy, males produce androgen, right? Testosterone is the main androgen. It doesn't equal androgen. It's the main or the most common androgen. Females have estrogen, right? Plus progesterone. That's in females only, right? Progestation means for pregnancy, and that's what's going to prepare the female uterus for possible fertilization and implantation. The other thing is females produce one gamete per month, while males produce about 500 million or half a billion of sperm. Most of those are going to be wasted, but it's one versus 500 million, half a billion. So here is the testes, and the testes produce androgen and sperms. For ovaries, it produces the equivalent, estrogen and eggs. But what else besides estrogen? Progesterone. So here is the ovaries, and it is called oocyte. How about we call it an egg? No, egg is not. We're doing reproductive now, so we have to be accurate. Egg is not accurate. Ova is not accurate anymore. It's called oocyte. Ova is a mature one, which is later on. So what the females have, is it the ova? Is it the eggs? No, it's called oocyte. Okay, it will not become an ova unless it is mature, which is fertilization. Okay, so this is a huge difference. It is oocyte. And hormones, estrogen, progesterone. And we talked about the female reproductive system before, including uterine tubes, the ovaries, the uterine tube, the uterus itself, including body, cervix, and then the vagina, right? And the vulva, which is the outside part, and so on. Uterine tubes. What's the other name for uterine tubes? Fallopian tubes. What's the function? It carry oocyte from the ovaries toward the uterus, right? And when do you think where do you think fertilization occur? It starts here in ovaries and it moves in the fallopian tube. Where do you think fertilization occur? Fallopian tube. Yes, it's fallopian tube, not the uterus. It's fallopian tube. So the oocyte is coming like this, and the sperm is coming from the other side, and they're going to meet in the fallopian tube. It's a common mistake to say uterus. It is not. It's in the fallopian tube. After fertilization occur, It's going to develop and move, move, move. It will be implanted at the end in the uterus. But where is the conception? Where does it happen? Sperm meeting the oocyte, it is in a fallopian tube. Okay, it is ultimately, but a zygote will move, not a fertilization. Okay, for males, we start in the testes. Do you remember these? Testes, which part of the testes that produce the sperms? Start with S. Semi, seminiferous tubules, yes, in the testes. And then it moves to the epididymis and stay a little bit. Why? You have an exam today. They can ask you about this. Why does it stay in epididymis for some time? For maturity. It matures in epididymis. Then it leaves the epididymis through ductus deferens. What the other name for ductus deferens? Vas deferens. And then ejaculatory ducts. Ejaculatory ducts come from ductus deferens plus something else. What is that something else? Seminal, the duct of the seminal vesicle. You have an exam today. You can see it. It come from, it's not ductus deferens will become ejaculatory duct. No, ductus deferens will join the duct of the seminal vesicle. Both of them, when they come together, they will make ejaculatory duct. And ejaculatory duct is going to pour into prostatic urethra. Yes. So it will go to the urethra, and from the urethra, it will be ejaculated out. So for males, you have three glands that you have to understand what each one of those does. Each one of those is a simple question or two. Okay, you will be asked about it. These are basics. You have seminal vesicles or glands, you have the prostate, you have the bulbourethral gland. Okay. Um, the testis is located inside the scrotum. So the scrotum is that skin surrounding the testis, or in which the testis is hanging there. Okay? It's a fleshy pouch that encloses the testes. And it is anterior to the anus and posterior to the base of the penis. Okay, so here I will talk about it the way the sperms are made. The sperms are made here in the testes. So this is number one, testes. After the testes is going to move. Inside the testes, there is a pathway I'm going to talk about. But generally speaking, testes to epididymis, its head, body, tail. What are the after the epididymis? Vas deferens or ductus deferens. And then look at this. It's going to meet this. What is this? Huh? What is this? Now, this is the ejaculatory duct. What is this? Seminal vesicle. Yes. And this is the duct of the seminal vesicle. Do you see the two here? This is the vas deferens meeting with the duct of the seminal vesicle. Both of them together go to the ejaculatory duct within the prostate. And then it's going to go to the prostatic urethra. What's after the prostatic urethra in males? Membranous urethra, and then spongy or penile urethra, right? Now, where are these glands that I just talked about that you have to know? Here is, we don't see all of it, but it's here. We'll see other pictures right here. This is the seminal vesicle. This is one. The prostate is one. And look at this. Can you see this? This tiny gland right here. This is called bulbourethral gland. Translation. Do you see all this purple? What is this? This. Spongiosum. Do you see this part of the spongiosum? You see how it is dilating in the back? This is the root. The root. The root have its own name. It's called the bulb. Okay? And you see this still coming from this part, right? Look, all of this is the same tissues, right? Bulb, the spongiosum itself, and what is this elongated part of the corpus spongiosum? Glands. So bulb and glands, both of them are parts of the spongiosum, yes. The bulb is the root. So why did you call this bulbourethral glands? Look at it. It's between the bulb and the urethra, isn't it? That's why we call it bulbourethral gland. How about the root? So what's the root of the spongiosum again? Bulb. What's the root of the cavernosum? This. Does anybody know what is the root here? It's called the crus. Crus is the root of the cavernosum. Like cavernosum and spongiosum, these are long erectile tissues, right? They have to be attached to the anterior pelvic wall, right? To be attached, you have to have a root. What's the root? Bulb and crus. So if you look at this, here it is. Look at this first. You see how the vas difference or the ductus difference, how it become dilated first before it join. This is called the ampulla. And then this is the seminal vesicle behind it. Its duct is going to unite with this part of the vas difference to make the ejaculatory duct. And here is the prostate, and this is the bulbourethral gland. Spermatic cord. What's the spermatic cord? We talked about this in the lab. Spermatic cord is basically a cord that takes everything into the inside of the body. Okay, it's a connection like a tube, a big tube between the inside of the pelvis on one side and the testis on the other side, or the scrotum on the other side. This big tube here, this is called spermatic cord. Why did you call it spermatic cord? Because it's the cord that contain the vas difference, which is conduction. The function is conduction of the sperms. And that's why we call it spermatic cord. So what do we have inside the spermatic cord? Vas difference, delivering the sperms, right? Plus arteries and veins and nerves and lymphatics. All of this bundled together is a communication between the testes and the inside of the body, the internal organs. Now, the testis is outside and the pelvic organs are inside, right? How does spermatic cord enter into the body? Do we have like an opening or something? The inguinal canal. This is your entrance and exit. This spermatic cord is going to go in and out through what? Inguinal canal. The veins, arteries, it's arteries, testicular arteries. The artery that go into the testes is testicular artery. The vein that's coming from the testes is testicular vein. But the testicular vein specifically, it makes a plexus. It's not just one vein that's moving like the artery, like this. No, it makes a plexus of veins. And we call it the pampiniform plexus. Okay? Since I'm saying the name, it means you need to know it. Pampiniform plexus. Pampiniform plexus. It's a plexus of the veins. Plus nerves coming from the lumbar plexus. It's called the genitofemoral nerve. Plus lymphatics. So we said that inguinal canal is this canal through which spermatic cord enter, right? And things enter and leave, right? If this opening become wider, not only the spermatic cord, it doesn't only accommodate the spermatic cord, anything inside can go out, right? If something herniated out of the body, we call that inguinal hernia. Inguinal hernia, did you understand the idea? If it is like this, and here is spermatic cord, barely go through it. That's normal. If it is like this, spermatic cord going, but you still have a space here, right? Anything can bulge out. We call it inguinal hernia. Look at this. This whole thing here, you see all of this surrounded by white here? They opened it. This is the spermatic cord, the whole thing. Testicular artery, testicular vein, but look how it looks like a plexus. And nerve supply and lymphatics. And this opening, can you see this opening right here? This opening here is the inguinal canal. There are two muscles that you have to know that are called, and they do almost the same job, almost. One of them is called cremaster, which is this one. Can you see this? We have a model like that. Cremaster or cremasteric muscle, it's the same thing. And this around the testes plus the spermatic cord. It goes up. And there is another one underlying the skin of, what do you call that skin here? Scrotum. So there is another one here that's lining the scrotum from inside that's called dartos. So how many muscles do we have here? Two, cremaster and dartos. Both of them do the same idea, and I talked about that. I will repeat again real quick. If you see a question, why do you have these? Simply, the testes to be working perfectly, it should be about 1 degree less than the inside of the body. The inside of the body is warmer. Do we know that? It is, of course, warmer, right? The inside is bad for the testes. The testes need one, it's 1.1, but it doesn't really matter. Okay, about 1 degree less than the body. So it should be close to the body. If it is cold, when it's hanging away like this, it's too cold for it. It's not good. Okay? It's like two less or three less than the body. That's bad. It has to be one. So these two muscles are going to contract and pulling the testes up, repositioning the testes. Now it's closer to the body and now it is warmer. Is that clear? How about if it is hot? Do you want to stay close to the body or do you want to move away? You want to move away. It's even more hot inside. So these muscles are going to relax so that the testes move away. The purpose or the goal is that you keep repositioning the testes depending on the temperature. The bottom line is I want to be 1 degree less than the inside of the body, than the core. You contract, you relax. It's all about the temperature. But at the end, you have to keep it 1 degree less than the inside of the body or less than the core body temperature. Do you know where the testes are located in fetus? Is it here? Or embryo? Is it here in the embryo? Where is it? It's up here, all the way up here. It's under the kidney. And then there is a ligament that pull it down. The ligament starts from here, okay, and goes like this. And the ligament will pull it down, pull it down, pull it down, pull it down, pull it down until it come here. Okay. So at birth, 90 something percent, it should be here. Occasionally, it takes a little bit some time. Like I told you guys before, right? So the doctor, the neonatologist or the pediatric doctor will come and check it out and see if both of them are here. If not, they are not going to interfere right away. They just give me 2 weeks or something and come back. If it goes down, it sometimes there is like a lag. The ligaments are not contracting the way it should. So if there is a lag, that's okay. After that, they have to intervene. Why? Because there is something called undescended testes. The testis didn't go, and it's not going to go. It's not going to descend. Okay? We call that cryptorchidism. Remember this. Undescended testes. It's called cryptorchidism. Cryptorchidism. You will see it. But it's called cryptorchidism. It means undescended testes. What's the problem? I just told you. Inside is warmer. Is that good for the testes to be inside? Of course not. Is it going to be functioning well? No. The other thing that's even worse, cancer. Heat, 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 heat. Cancer come from anything that's abnormal, right? Consistent or chronic exposure to something that's not normal can lead to cancer. So that's why it has to go down to the extent of they can intervene and they can pull it down. Worst case scenario, they can get rid of it, but they can't leave it anyway. And it's if it is the case, most of the time it's unilateral. Okay, so surrounding the testes, there are tunics, and tunics means layers. There is tunica vaginalis and tunica albuginea. Tunica vaginalis is 2 layers, visceral is inner and parietal is outer. And then after those 2 layers, I will show you the picture. There is the dartos muscles and cremaster muscles, and I talked about those already. And here is the purpose that I talked about. Okay, you can reposition according to the temperature. Is it hot? Is it cold? And so on. The other one is tunica albuginea. So we have 2 layers surrounding the testes. Tunica albuginea and tunica vaginalis. It have nothing to do with the vagina. We're talking about females, but it is just a term that's giving to this layer. Now, the inside of the testes, if you look inside, you will see lobules, 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 lobules that are separated. And we saw that in the lab. Do you remember this blue testis model that was opened, and you see these golden tubules separated from each other. The separation um, is septum that's separated into loops. And if you look inside the loops, you see seminiferous tubules. And these seminiferous tubules is where we make the sperm, a very common question. It's not enough to say that we make the sperms in the testis. Everybody know that, right? If you ask a child, they will tell you the same thing. But anatomy and physiology, where is it? Seminiferous tubules, specifically. After the seminiferous tubules, we're still inside the testes. There is something called retitestes, straight tubules, efferent ductules, and then the epididymis. Remember, I showed you the picture and I told you it will go from the testes to the epididymis, right? And stay sometime for maturation. But does it go from the testes to the epididymis just like that, jumping? No, inside there is a pathway. So look at this picture and it will show you what I talked about. Here is the inside of the testes. Look at these lobules. Here is a lobule, and there is a septum lobule. The purple is a lobule, and the yellow is a septum. Okay? What do we have inside the lobules? This seminiferous tubules. What's the surrounding? Tunica vaginalis and tunica albuginea, and then tunica vaginalis. Okay? Albuginea is the inside, vaginalis is the outside. So what's the pathway of the sperms? It goes from the testes to the epididymis. It doesn't jump. From the seminiferous tubules to this, which is called retitestes, and then, from the seminiferous tubules to the straight retitestes, efferent ductules, and then the epididymis. So it has to go through those before going to the epididymis. In the epididymis itself, the epididymis looks like this. This is the testis, it's hanging above it like this. Okay? Head, body, tail. And what's after the tail? Vas deferens or ductus deferens. Okay. Inside inside the testes, in between these tubules, there are cells that's called interstitial endocrine cells or Leydig cells. You need to remember the cells. You have to remember the cells that makes the sperms and make the testosterone. Not only the testes, that's not enough. Not only the seminiferous tubule, you need to know seminiferous tubules, that's okay. But cells, to be more specific. So which cells make androgen? It is interstitial or Leydig. Interstitial or Leydig. You should remember that. So the testes, specifically seminiferous tubules, this is where we make the sperms. The sperms go to it follow the pathway that I talked about, and then go to the epididymis, staying for a little bit in the epididymis. Every one of those is a question, right? What's the function of the epididymis? What's the function of the vas deferens? What's the function of the ejaculatory duct? Each one of those is an obvious question. Okay, it can be luxury or lab, both. That's why I'm saying pay attention, pay attention. Okay? It's not only for the lecture. So here it is. It go to the epididymis, which is head, body, tail. But in the epididymis, it is for maturation. Head, and then it go to the body, and then it go to the tail. Here it is. Head. The head is the part that's hanging above the testes. And then body, and then the tail will end up going to the vas deferens or ductus deferens. And the ductus deferens will go to the body inside what? Within. How does the vas deferens enter into the body? Within what? It inguinal canal, but what is the bundle that take it in? Spermatic cord. Yes, spermatic cord. What else do you have in the spermatic cord beside the vas deferens? Blood vessels, artery and veins, nerves and lymphatics. All of that together is going to go up. Here it is. This is actually how it looks like. Testes, head, body, tail. Here is the vast difference. And the whole thing here is a spermatic cord, including everything else that we talked about. And the epididymis is for maturation. This is one. Plus, it adjusts the fluid that's coming from the seminiferous tubules. Did I mention that testes, I mean the sperms, we make it in the seminiferous tubules, right? Sperms in the seminiferous tubules, how does it move? The sperms, in order to move, it has to move in fluid, right? It cannot move just like in the tissues like that. It has to move in a fluid. So this fluid carrying it, when it go to the epididymis is going to allow the sperms to maturate, plus monitoring and adjusting the fluid. And if there are bad sperms, it's going to be recycled as well. So, but the most important of all is maturation, but also adjusting the composition and recycling damaged sperms. Ductus difference is goes within the spermatic cord, and it goes all the way within the spermatic cord and enter the body through inguinal canal. The parts that become dilated before joining what? Act of the seminal vesicle to make ejaculate. Are you following with me? Ejaculatory duct. So it has to dilate like this first, which we call it ampulla. After the ampulla, it joins the duct of the seminal vesicle, making ejaculatory duct. So what's the function of the ductus difference? Delivering the sperms, of course, within the fluids. Delivered all the way to ejaculatory duct. Male urethra is three parts, and I already talked about that. It's about 20 centimeters, prostatic, membranous, and spongy, and we talked about that. Accessory organs, each one of those is important for the male function or for making the semen. What's a semen? Is semen different than sperms? Semen is sperms plus secretions, fluids. The fluids come from three glands. Two of them are the main ones, and one is just a little tiny bit. Seminal vesicle and prostate, these are the big ones. Most of the fluid come from this. Okay? The bulbourethral gland, just a little bit, and it's more of lubrication. Okay, bulbourethral gland, is it part of the semen? Very tiny portion, but it work in the beginning as it goes like during sexual stimulation. Some fluids go to the glans penis for the purpose of lubrication and making the sexual intercourse easier. Okay. Uh, so look at this. This is the vas difference. What is this? Ampulla. It's called ampulla. And what is this? Seminal vesicle. You see the join to make ejaculatory duct that enter into the prostatic urethra, membranous urethra, and then spongy urethra. Look at the bulbourethral gland. During sexual stimulation, it releases a little bit without anything else. Just a little bit go through this. And again, it's responsible for lubrication. Each one of those functions is important. Okay? So why do we have all these accessory glands? To make the semen, right? Which is supplying the fluid. Is that fluid just a fluid just for the sperms to be located in a fluid? No, it's not. This is not enough. It produces certain things within the semen to activate the sperm, nutrients to the sperm, moving the sperm, buffering the fluid. Do you know what's buffering? We just finished 27. What's buffering? Regulating the pH, neutralizing the pH. It shouldn't be acidic. Okay, if it is acidic, it's going to add alkaline. Did you get the functions? So seminal vesicle is the first one. And this is about 2/3. This is the major one. Remember this. Major one is the seminal vesicle. Prostate is like third, and the bulbourethra is like 1%. So the vast majority come from seminal vesicle. Remember this. Come from seminal vesicle. Ejaculatory duct, we take it from there after the ampulla, joining the duct of the seminal vesicle, make ejaculatory ducts, and goes into the prostatic urethra. The prostate is number two, and it's about thirds come from the prostate. Okay, the fluid of the prostate is slightly acidic, but the seminal vesicle fluid is alkaline. Remember this. It's alkaline, so it's going to neutralize it. So is the semen alkaline or acidic? Alkaline. Yes, it end up being alkaline. It has to be alkaline. If it is not alkaline, sterility. You cannot have kids if it is not alkaline. For a reason, I will mention it. Okay. If inflammation happen to the prostate, we call it prostatitis. Alburythral gland, this is like 1%. Function, I just mentioned it, lubrication, secrete a little bit to the glans penis, the tip of the glans penis specifically, to make the penetration easier. Plus, it also neutralizes the acidity. Now, why are you saying like stressing on this point? Why alkaline? Alkaline, like seminal vesicle is alkaline. Plus fructose, alkaline and fructose. You should remember this. The seminal vesicle, you have to remember three. It's the main component, 2/3, that's one. Alkaline, that's two. Fructose, that's three. Fructose as a nutrient to the sperms. Otherwise, it's going to die. Now, why I'm mentioning seminal vesicle is alkaline. Bulbourethral gland is also neutralizing. Why I'm saying that? Because remember, the male urethra is serving two purposes, right? It's for urine and semen, right? Urine by nature is acidic. If you remember from the urinary system, the urine is acidic. Don't you get rid of hydrogen in the urine? That's normal. So the urine is acidic. If the sperm is exposed to acidity, it's going to die. Not only that, the vagina is acidic. That's normal. Okay? There are, I think we all heard about the lactobacilli. Did you hear about the lactobacilli? Lactobacilli is normal inhabitant. We know that there are bacteria that normal in the skin, normal in the mouth, normal in the vagina, in the anus, and right? We know that. There are organisms all over the body, and it is normal to be there. This is normal habitat. So the normal habitat of the vagina is lactobacillus. Lactobacillus will make lactic acid. So the vagina is acidic. So if the sperms, these weak sperms, if they go through the urethra, which is acidic, and then to the vagina, which is acidic, it's going to die. Okay? And this is one of the causes of infertility. It's not alkaline enough. Okay? That's why the secretion is alkaline. And bulbourethral is also helping to neutralize the acidity of the urethra itself. It's slightly alkaline as well. So at the end, the semen is alkaline. So when the semen go to the vagina, it will find the vagina acidic, and it will neutralize it so that the sperms doesn't die, and then it will make its way up vagina, cervix, uterus. What's the end? Where does it go? To the fallopian tube. Did you get this pathway? Vagina, cervix, uterus, out to the fallopian tube. This is a normal path. In order to do that, it has to be strong enough. To be strong enough, nutrients um, the pH should be slightly alkaline. So here is the semen. The semen is about 2 to 5 milliliters. This is a normal ejaculate, and you should remember these numbers. 2 to 5 milliliters. This is normal. And the sperm count that you need to remember is from 20 to 100 per ejaculate, like the whole ejaculation. No, we don't measure it per ejaculation. If there is a couple having like an issue, like infertility, or the lady cannot conceive or something, the first thing is, let's see the male first, right? Give us semen sample, and let's look under the microscope. In 1 milliliter only, it should be 20 to 100 million. So it's 10 million okay? No, it's not okay. 10 million. No, it's not okay still. Okay. Because a lot of those are going to be weak and die. So it has to be within that number that you have to remember. 2.5 milliliters, 1 milliliter of those two to five, it should contain about 20 to 100. Less than that is abnormal. Even if it sounds a lot, it's not normal. So here it is, seminal fluids will have fructose coming from seminal vesicle. It should also contain prostaglandin. Everything here have a reason. What's the prostaglandin? Contract the smooth muscles. We know ejaculation is the semen is pushed out into the vagina, right? This is physiology. How is the semen pushed out? You have contract the muscles to push it out. Peristalsis, right? What stimulate the muscles? Prostaglandin. So you should remember fructose, prostaglandin. Fructose and prostaglandin. Prostaglandin stimulating the smooth muscles to contract to push the semen out. At the end, the semen will contain sperm, the fluid, plus some enzymes. The fluid is coming from the three glands that I talked about. And the enzymes are protease. Seminaloplasmin is antibiotic. See all these? Fructose for nutrition, prostaglandin for movement, alkaline to protect against acidity, antibiotics, which is seminaloplasmin, to kill if there are like small number of bacteria, it can kill it. So all of these together are going to help to make the sperm healthy. But if any one of those is not normal, it will be an issue. Now, the penis itself is the outer or external reproductive organ of males. And this is for sexual intercourse and also for urination, right? It has a root, and I told you guys the roots, right? What's the root of the spongy? Bulb. Bulb urethral is a gland. What's the name of the root of the spongy? Bulb. And what's the name of the root of the cavernosa? The crus. Crus. So for the penis, you have a root to attach it to the body wall, and then you have the body and the glans. The glans is part of which one? The glans penis. Is that part of the spongiosum or cavernosum? Spongiosum. It's an extension from the spongiosum. So look at this. These colors are like that for a reason. Which one is this? Pink? Spongiosum. So look at this. This is the bulb, right? And the urethra enter from the bulb, but the bulb is the root. And here is the spongiosum one or two? One that carry one urethra. And look, when it go to the front, it's uh, enlarge and make the glans penis. Look at the cavernosa on the other hand. It's two. And look at the root. Here it is. Crus. This is the root of the cavernosa. What are all these three? Why do we have these three? Two cavernosa and one spongiosum. Why? What's the purpose? Erectile tissues. Yes, erectile tissues. Okay. The three are erectile tissues, but the spongiosum, plus being erectile tissue, it's carry the urethra. Okay. Um, I talked about um, the prepuce before, or the foreskin. Remember both names, prepuce or foreskin. And this is the last part or the terminal part of the skin surrounding the penis. And I told you guys that it can be cut away, and we call that circumcision, or it can be left alone. So it's the I mentioned that doctors always ask the parents, do you want to do circumcision or not? If that's the case, circumcision is cutting the front part or the distal part of the foreskin. And the rationale is if it is there, it can hide organisms and pathogens underneath. So, of course, it is everybody have a choice, but they give the option to everyone. So here is circumcision is removing part or the whole foreskin to prevent, here it is, infection and cancer. Erectile tissues, we have those three. Two of them are cavernosum and one is spongiosum. Inside of them, you have arteries and veins. What happened exactly? Here's what happened. These erectile organs, right? Normally, it is flaccid and the size is small, right? Flaccid, soft, small. Artery goes in, vein goes out. Erection occur by the arteries dilating. So more blood goes in. Veins constrict. So the blood is trapped, right? And that will increase the size and uh, make the erection. It make it more what they call it erected, okay, and increase the size to be ready for sexual intercourse. So again, it becomes stiffer. That's what I was trying to say, stiffer. And assume the upright position will be straight, stiff, and big. This is what erection is all about. So again, arteries, blood move in, veins constrict, trapping it inside, and this is erection. So here are the two cavernosa, and I mentioned the root is called the crus, and it extends all the way to neck of the penis. Does it go to the glans penis, cavernosa? No, it ends at the neck. What's the neck? It's between the shaft and the glans. There is a neck there. Okay? So it only go to the neck. Spongiosum, on the other hand, it's only one. And this is the one that contain um, the urethra. Sorry, the urethra. Okay, look at this picture right here. All of this is the same color. Again, this is the bulb. This is the shaft. The neck is right here. Okay, you see the purple or the violet end here, right? This is the neck. It's between the glans and the shaft. So root, shaft, neck, glans. Neck is where the cavernosa ends. And here is the root, crus, and here is the root, is bulb. And here is the foreskin. What's the other name for the foreskin? Prepus. Prepus. And if they cut it like here at the neck. Here is a cut section. This big one here and this big one here. Two of those are cavernosa. Do you see the artery inside here? These are the arteries that's bringing the blood in. And the veins right here, the veins are going to take the blood out. Again, during erection, the veins constrict. And here down there, the pink is the spongiosum and the urethra is inside of it. Uh, spermatogenesis. Spermatogenesis, generating the sperms. Body cells, the body cells contain, are called diploid cells. Diploid is coming from double. Okay, it's two sets of 23 chromosomes. This is the normal body cells. Okay, so this is the skin cell. It's a diploid, contain two sets of 23. It make another skin cell identical. Same thing. Are we following? Diploid making diploid. 46 making 46, 46, 46, 46. These are the normal body cells. And this is called mitosis. You start with 46, 146, you make two. Each one contain 46. And you make four. Each one contain 46. 8, 16, 32, and so on. 30, 46, 46, 46, 46. All of these are diploid. This is not the case here with the gametes. You want it, the gamete, and what's the gamete? Sperm or oocyte. You want it to be haploid. Haplo is half. Dipole is double. Clear? Two sets versus one set. Two sets, each one is 23, so it's 46. This is the dipole. Dipole, two, double. Haplo, half of that, so it's 23. So it starts with the diploid. And the diploid is two sets of 23, and these are always identical. Number one here and number one here is exactly the same. Two, two, three, and all the way until 23, identical pair. So there are the steps. And we did that in biology. You guys did biology, of course, right? General biology. So you probably remember how it's going to like come together and disappear. The nucleus disappear, the nucleolus, I mean, disappear, the wall disappear, and you go to prophase, metaphase, anaphase, and telophase. You guys still remember that? This is mitosis. Meiosis is what's important here because mitosis is every cell in the body except gametes. So meiosis is gametes. That's it. And the purpose is to make haploid gametes, N, one N only, one set. Two N's is diploid. Okay? So the purpose is to make haploid cells. So does the sperm have a diploid? Is a diploid or haploid? Sperm. Haploids. Same for the oocyte. It's haploid. What's haploid? 23 or 46? 23. It's one set only. Okay. Why is that? Because the purpose is you end up having a sperm with one set. You have an oocyte with one set. So when they merge together, you end up having a normal cell. Make sense? So it will be 46 at the end. So the zygote will end up containing 46. Here is a parent cell, and it go to meiosis, but meiosis is different steps. There is meiosis 1, go through prophase, metaphase, anaphase, telophase, and then you repeat it again. And you end up having 4. Here it is. 1, meiosis, prophase, metaphase, anaphase, telophase, you end up making 2. You don't want to make 2. You want to make it 4. Clear? So what should I do now? Meiosis 2. Each one of those, prophase, metaphase, anaphase, telophase, you end up having 4. Clear? So you end up having 4, not 2. That's why you do it twice, which is meiosis 1 and meiosis 2. But you end up having 4 what? Haploid or diploid? Haploid. So you duplicate the cells first, you do the mitosis, and then in meiosis, you're going to make it half, meiosis 1 and meiosis 2. Okay, what happened in meiosis? Prophase, metaphase, anaphase, telophase. Do you still remember that? Prophase, nuclear membrane disappear, nucleus disappear, so everything is like loose inside the cell, and then metaphase arranged around the equator, and then you pull them apart, right? Duplicate and pull them apart, which is anaphase, and then they move into two cells, and we call that um, the cytokinesis. So meiosis 1 go through all these steps and ending at the telophase. Prophase, metaphase, anaphase, and telophase. Telophase is the last one before the cytokinesis. So you end up producing 23. There is a stage before this or in between that's called interphase. It doesn't go meiosis 1 to meiosis 2. Meiosis 1, interphase, and then go to meiosis 2. This phase in between is called interphase. And then you go to meiosis 2, same thing again. Prophase, metaphase, telophase, but you call it 2. So this process of spermatogenesis start in puberty and ends at around 70. Okay, this is like an average. What do you call it when females stop making oocytes? Menopause. Does males have something similar to that? Andropause. Andropause. It's not exactly the same, but it's called andropause. Um, so you go through mitosis first, and then meiosis, and then spermiogenesis. These are the steps. The stem cells, you need to know this. Stem cells is called spermatogonia. Spermatogonia. This is the stem cell. This is the one that contains the 2 sets. This is the diploid. This is the beginning, the stem, the origin. It's going to divide by mitosis to make primary spermatocyte. After primary spermatocyte, you have secondary spermatocyte. Okay, here is primary spermatocyte, and this is mitosis. So it starts with 46. What do you have in a primary spermatocyte? 46. Still. Secondary spermatocyte, 46. And then the spermatide, which is immature gametes. And then the mature one is called sperm. Sometimes we call it spermatozoa. And this is the one that's actually leave the seminiferous tubule, go into the fluid, and move to, it will move ultimately to epididymis, right? Epididymis. So spermatogonia, primary spermatocyte, secondary spermatocyte, and you end up having spermatides, and now it's half. Are we following? Spermatogonia, primary spermatocyte, secondary spermatocyte, and then spermatide, which is haploid, and then the sperms, which is now we are talking about meiosis too. At the end, listen, at the end, you started with one stem cell, spermatogonia, and you end up having four sperms. All four are okay. All of them are working fine. Females is different. You will see it. Female, we start with one, you end up having one functional. Is that clear? Males, you start with one, you end up having four functional. Females, you start with one, you end up having one. Why? Why it's not four? It is actually four, but three of them dysfunctional disappear. So the bottom line is you start with one, you end up having one functional, because the other three, it's there, but not functional. So spermatogonia, primary spermatocyte, secondary spermatocyte, spermatide, and sperm. This is more details about the process. You can read it if you want to, but here's what I want you to know about the sperm. This is important here. The sperm consists of head, okay, middle piece, head, neck, middle piece, and tail. Head, neck, middle piece, tail. You need to know the function of each one of those or characteristics of each one of those. Head. Head contain the chromosomes, which is the most important thing, right? Chromosomes. The head is what contains the 23 chromosomes, okay? Plus, look at this here at the tip. There is something called acrosome. Acrosome is like the head or the tip of the head. Can we see that? The whole head is like this. And this tip here, just this tip, the very front part, this is called acrosome. What's the importance of that? The acrosome contain acrosomal enzymes. So when the sperm goes like this, and when it's finding the oocyte, how can you enter? You have to dig a hole, right? You have to open. How to open? Enzymes, acrosomal enzymes. These enzymes will be released, making an opening so that the whole sperm will enter? No, just this material inside. The nucleus is the one that's going to enter into the oocyte, and we call that the zygote. This is fertilization. Are we following? So what facilitates the process of penetrating into the oocyte? Acrosomal enzymes. Okay? Neck, it's just a neck. Middle piece, mitochondria. You have to remember this. The middle piece contains mitochondria. It's easy to remember because M goes with the M. Can you remember that? Middle piece, mitochondria. Middle piece, mitochondria. And then the tail is just for movements. So the head contains two things, right? Nucleus, which is the chromosomes, plus the acrosome. Spermiogenesis is the last step of the spermatogenesis in which the spermatide will become a sperm. So spermatogenesis is all the way until the spermatide. The last one alone, spermatide to sperm, we call that spermiogenesis. Did we understand? Spermatogenesis is all of this, from spermatogonia all the way down. The last step only, spermatide to a sperm, we call that spermiogenesis. Last thing here is Sertoli cells or the nurse cells. These are the cells that are important for the spermiogenesis. Remember the cells that I talked about before? I told you I'm going to ask you about the cells specifically, right? This is the last thing. What are the cells that are important for hormone, testosterone? Not the nerve cells, for the hormones. Interstitial or Leydig? Interstitial or Leydig? How about for the sperms? Sertoli or nurse? You have to remember this. The name of the cells. Okay? And we're going to stop here.